Thank you so much for joining us today. My name is Andrew Olson, and I will be your host and moderator for today. So our next speaker is a great guide for us and is very familiar with this topic. Dr. Eric Seiser is one of our outstanding senior field application scientists with Qiagen Digital Insights, and he's located in the DC metro area. Eric has a master's degree in bioinformatics and a PhD in functional genomics. His previous research focused on pediatric cancer biology at the Children's Hospital of Philadelphia, the comparative oncology of genomic alterations in dog cancers at NC State University, and cancer pharmacogenetics at UNC. As you can see, his presentation is titled Translational Genomics, Putting the Pieces Together from Variant Calling to Biological Insights. And with that, I will hand it over to Eric. Eric, are you with us?
Yes, Andrew, and
awesome.
Thank you for the introduction. Good afternoon, everybody. So as Andrew mentioned, my goal today is to demonstrate to everybody essentially how we can move from a scenario where we're starting off with FASTQ files from sequencing. We're going to call variants, filter and prioritize variants, and then define some biological interpretations from some specifically loss of function variants in a cohort of samples. So before I begin, again, a quick legal disclaimer, Kaijin products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. So for today's exploration, what we're going to focus is on a specific case study of interest. And this case study is focusing on Lynch syndrome. As many of you may be familiar with, Lynch syndrome is a hereditary condition characterized by high risk of various cancer types. And typically it manifests in the context of germline variants in one of the many DNA mismatch repair genes. So in particular, we've identified a study here that is looking at surveying for pathogenic variants in hereditary cancer syndromes with patients that have Lynch syndrome. So we're doing some germline NGS sequencing. In this study in particular, the authors utilized a targeted panel, and their focus was on just identifying and trying to highlight some variants across a cohort of 84 samples, which may have variants of relevance for disease pathogenesis. So in our context today, we wanted to choose this study in particular because we're going to illustrate how not only can you utilize your own data, but you can also utilize public data to do some translational research as well. So for today, our focus is going to be on three main areas. First of all, data analysis, how we can efficiently retrieve and process FASTQ data with accurate variant identification. Secondly, interpretation, how we can take a set of variants and rapidly assess both sample variants for disease relevance and pathogenicity. And lastly, for the discovery side of things, how we can combine results from a cohort of individuals and contextualize variant biomarkers with other public data sets. To do these three steps, we're going to utilize three of Qiagen Software products. One is for analysis out of CLC Genomics Workbench. For interpretation, we're going to use Qiagen Clinical Insights Interpret Translational. And for discovery, we're going to utilize Ingenuity Pathway Analysis. And so for the first product, CLC Genomics Workbench, essentially what this tool represents is the ability to process raw NGS data and get to various endpoints. So in our focus for today, we're going to be talking about DNA-seq analysis on a targeted panel. And in that context, the endpoint would be calling variants. But the software platform also supports other research endeavors. If you're generating, let's say, RNA-seq data, you want to look at gene expression or fusion information, the platform can support that as well. Now, in this particular slide, what we're going to first start off by illustrating is actually the endpoint of where we want to get to within the Genomics Workbench. So on my screen in particular, towards the center, you'll notice two key areas. The first one at the top is essentially a visualization of a particular variant of interest within my data set. So at the top, I have a pileup of all the reads identified at this variant locus that gives me some degree of looking at the actual sequencing evidence for a variant of interest. Next line here, I have the focus of a particular deletion that we've identified. And lastly, on the bottom here, we have an annotation to the amino acid changes that would occur based on that specific variant. Concurrently, at the bottom of the screen, what we have are a list of all the variants that have been identified in a given sample. And we have a table of annotation for those variants. The type of variant, various metrics with regards to coverage and frequency within samples, gene level information, and predicted amino acid changes. So ultimately, through the use of workflows, as we'll see today within the software, we can very quickly and easily get to accurate variant calls, in this case for germline samples, or also in the context of cancer research, if you're working with somatic samples as well, get to those variants of relevance in your samples very easily. Now, to do this, we're going to do a couple of key steps within the software itself. The first one being to download reference information and import our sequencing reads. Now, again, this can be in the context of, as we're going to do today, utilizing public data, or conversely, if you have your own sequencing data, importing that as well. Then when we talk about specific targeted sequencing, obviously there are regional information that we're going to want to bring into the software as well. So we're going to want to import our panel-specific information. And then from there, what we're going to discuss is the DNA-seq workflow itself, which will take us essentially in a single dialogue to do all the processing necessary to move from FASTQ files to called accurate variants. And then from there, we can parlay into interpretation. Now, for today in particular, because we are working with germline sequencing, just some of the steps that will be included in the workflow that we're going to utilize is there'll be initially trimming of the FASTQ files to make sure we have accurate and high quality sequence to begin with. Then we'll go through the steps of mapping to a reference genome and various alignment, refinement steps. The next step will be to call variants and then annotate them with transcriptional information as well as variant database information and do a number of filtering steps. And then from there, we'll cover it generating various QC reports. In particular, when we talk about targeted sequencing, coverage reports are oftentimes very useful in making sure we have enough sequence to accurately detect variants. And then lastly, kind of exploring the variants themselves. Now, just a quick side note that if we were to be running the same process on somatic samples, that there are scenarios where within our workflows, we also have additional capabilities that you might want to employ, such as identifying copy number variants, calculating tumor mutation burden, and also interrogating microsatellite instability status as well. So really, the software itself is a very flexible platform. The workflows that we're going to discuss today are those that can be utilized out of the box with the software, but they also offer a degree of customizability as well. So really what I want to walk you through within the software briefly today is the key steps to generate our set of called and accurate variants through CLC Genomics Workbench. And that's going to involve, again, a few main steps. First of all, downloading necessary reference information. Downloading our sequencing data itself. Again, we're going to pull data from this study on Lynch syndrome, so we're going to pull it from the sequence read archive. If we were to import our own samples, we would do that step as well, and then also bring in targeted information for our panel of interest. And then very lastly, running a workflow to call our variants and get our endpoint to transition into interpretation. So what I'd like to do now is to switch over into the Genomics Workbench. And again, I'm just going to quickly walk you through some of the steps I just mentioned. So this is the primary interface of the software, and we're going to show you how you can quickly and easily accurately call variants using this interface. Now, for everything that I'm showing today, I've actually run the analysis locally on my machine. So we can, in fact, accommodate local analyses on a laptop or a desktop. When you begin to start interrogating larger sets of data, larger cohorts of samples, there are also various deployment options outside of a local installation, such as employing a server for added compute power and also deploying workflows on the cloud as well. So really how you utilize the Workbench is very flexible, but ultimately, regardless of your deployment, you're able to actually run the analysis directly through this desktop interface. So as I mentioned, with regards to any NGS-based analysis, the first step we're going to do is capture the reference information. And if you direct your attention to the top right corner of the screen, the CLC Genomics Workbench makes this step very easy we simply need to click on References. And what we've done here is Qiagen has actually curated sets of reference information that will be useful for the various workflows that we offer in the software. And for today's example in particular, I'm going to utilize this HG19 RefSeq set for references. Now, through a singular download here, what I will capture is the genome sequence itself. In addition to that, information with regards to the transcriptome in the context of RefSeq. And then, as you can see here, other third-party database information, dbSNP information, ClinVar information, and information from 1,000 genomes as well. So by simply clicking this download button, I will generate all the necessary reference information that I need to run my analysis. With that step already done, the next step that I would do is one of two things. If I'm going to work with my own samples, I'll show you how we can import those in a minute. But again, for our example with Lynch syndrome, we're utilizing public data. And very nicely, again, if you direct your attention to the top right corner of the screen, the Workbench has a built-in downloader for pulling FASTQ information from the sequence read archive. So very simply here, I can click Download, Search Reads in SRA. And at this point, what I would go ahead and do is I can pull in a specific accession number for a sample, or in my case, I can paste in here the project accession number, simply click Search. And what I will capture from this search, again, recall within this study, I mentioned that there are 84 samples within this cohort. So now we have all of these 84 samples listed here. And for any of them, we can first and foremost select them. We can show the metadata for these samples. So we have now a table for the samples I selected from the study, a set of descriptors, again, various accession information regarding to the project. So here you can see we have a 94 gene panel that we're working with. And then in addition to that, as I would scroll further along this table, we will get sample specific information, the isolate, the age of the sample, the sex, and any additional details that might be included within this study. So that gives us some context to what these samples are. Now, for any sample of interest, I would have the ability simply to select that sample or select multiple samples and simply click Download Reads and Metadata here. And that will get the data directly into my software. So there's no need to move externally to capture my sequencing data from a public repository. Now, with that simple step out of the way, I just want to diverge a second and talk about quickly how, if you had your own sequencing data, you could bring that into the software. And in this scenario, again, it's a very easy process where we would go to the section here for import. And if we, let's say, had Illumina sequencing data, we could select the option here for Illumina. We can take in either your single end or your paired end reads and very simply get that into the software. The other thing that we would do with this import as well is that we can bring in information with regards to any primers, let's say, that were used on your particular panel of interest. And in addition to that, we can bring in track-based information or the bed file noting our targeted regions. So again, very simply through a couple of clicks in the dialog, you can get your necessary reference information and both your sample and experimental specific information into the software quite easily. Now, when we bring this information into the software, you'll notice to the upper left-hand side of the software, we have this navigation area. Essentially, this represents the directory structure within the software. And you'll note here in today's example, I have a folder created for Lynch syndrome data. And under that, one of the FASTQ files that I've downloaded from the sequence read archive. And what I can do is I can open that file up. I can look at the FASTQ file. Again, lots of information here. So this is now a scenario where at this point we're ready to process the sample itself. And to do so, where we will refer to is the bottom left corner of the screen. In particular, in this section, we have sets of individualized tools, such as tools for mapping or variant detection or various filtering or annotation steps. But what I want to highlight today are these ready-to-use workflows. So essentially, these represent a scenario where we have built out pipelines for analysis that do all the necessary steps to process your data, and you simply have to go through a singular dialogue to process one or more samples. Now, in this context, when we talk about variant detection, tied to Dan's talk earlier, we do offer actually pre-built optimized workflows for the various QIAseq panels that are offered through Qiagen. And then in addition to that, we offer various workflows in place for whole genome sequencing, exome sequencing. And again, in the context of what I'm doing today, we have a targeted panel that we're going to look at for identifying germline variants. So outside of this, again, we would have various tools in place for, again, doing things like mapping reads to reference, variant detection, what have you. So really, you have the ability to utilize a workflow, process a sample in a standardized fashion very quickly, or do more exploratory analysis, process the data in a stepwise fashion using individualized tools. Now, just to show you how easy it is to both interrogate a workflow and also run a workflow, I'm going to pull up a copy of the workflow that I've created today. And when we look at these workflows, essentially, they can be distilled into three main types of things. The green boxes represent inputs for the analysis. The three-tiered boxes represent specific tools that are being run. And the blue, maybe purple colored boxes, they represent outputs from the workflow. And now to quickly kind of review what's going on in my workflow for today, we have initial trimming steps that we do. We have mapping steps for the alignment. There will be then various steps here that we do to refine the alignment. So we'll do structural variant calling, a local realignment. And then from there, we'll transition into a variant detection step. Now, everything that we would do downstream of here would essentially relate to various tools that are in place to both annotate and filter your variants, such that when we get to our set of called variants as a result of this workflow, that we have accurate data being reported. Now, one final thing to note with these workflows is that, again, they are flexible. You can build your own workflows, you can add tools to existing workflows, and you can also modify and lock down any parameters of interest within a workflow. So at this point, if I'm ready to run the workflow, very simply what I would do is come to the bottom right corner here. I would click the run button. And now we'll essentially walk through a dialogue where, let's say, we can take one of our samples of interest, or I could take multiple samples and I can batch them. And then all the available parameters that we need to enter can be selected throughout this dialogue. And in this case, many of our pre-built workflows actually require very limited interaction. You can modify that to expose any parameters you want, but here I would essentially choose my reference data in this step. The next step, if I had any pseudogene information for trimming, which I don't, I would import that here. And essentially everything else in the workflow has been optimized already such that I don't need to specify any additional parameters. So at this point, what I would do is just choose a location to save my results, click finish. And again, I'm running this locally. These samples, a targeted panel, so smaller data, ran well under an hour. And again, if you have larger cohorts of samples or larger data overall, you can explore our both server-based options and our cloud engine-based options as well. So at this point, I've gone ahead and I've run the workflow in advance just for time purposes. And let's take a look at some of the outputs. So in my navigation area here, I've designated a folder for the outputs of the analysis. You'll see there is a subfolder here capturing different QC reports. So in particular, in this scenario, I might have a general QC of my FASTQ sequence, information with regards to trimming and mapping and structural variant detection. But what I want to highlight very quickly is just a coverage report. Again, when we're doing variant detection, understanding coverage within our samples is very important. And when I open this report, what I'll see very nicely is that, in fact, we have high overall coverage, over 600 reads per sample. The median coverage is very similar. And when we look at the percent of targeted regions that have a certain threshold of coverage, again, just about all of our regions here have a coverage of 30 or greater, and on average, 600 reads per locus. So this gives us some context to know that we have appropriate sequencing depth to hopefully get a good sense of the variants within our sample. Now, there are other various pieces of output for this analysis. Typically, you would find that under track data, you can get the mapping information, the pre-filtered variants, and a number of other things. But really, the key endpoint that I want to show everybody is this genome browser-based view. And this is a very nice integrated view where, again, similar to what I showed on the slides, at the bottom of the screen here, what we have is a table of all the called variants. Within this table, again, we have positional information, zygosity, coverage, frequency, metrics related to variance confidence from the caller. And towards the end of this, information relating to, again, annotation of the specific variants identified. So for any of these variants, they're dynamically linked to the genome browser-based view above, such that I could come in the table, I could focus on this particular deletion. It's in the MLH1 gene, so a gene of note for Lynch syndrome. And as I click on that row within the table, now what I'll see is a view of the mapping at the top here that denotes coverage. I'll see a positional information of the variant allele itself here. So we're dealing with the deletion of 2 nucleotides. And then as I would scroll down, we can see that it's within a targeted region using this blue bar here. And then also look at the expected amino acid change based on this variant. So ultimately, within the workbench, through a couple of easy steps, I can import all my necessary reference information, my sample information, and very quickly run a workflow that accurately calls variants in my sample of interest. Now, at this point, what I'd have the ability to do is export the data, and we can export this data in a number of different formats. We can export a table of variants or a VCF file. We can export PDFs for any variant level reports. And also of note, within the software, we actually have a built-in exporter to transition our variant calls from CLC Genomics Workbench to Qiagen Clinical Insights Interpret Translational. And in fact, let's go ahead and jump back into the slides. And at this point, we can talk about our next step, which is variant interpretation. And so for this process, typically our starting point is going to be a VCF file where we have a large set of variants. And our goal is to essentially utilize information to both filter and prioritize variants in our sample or our cohort of samples. And to do this, we're going to rely on the Kaijin knowledge base. And essentially what this represents is a massive effort of curation. As Beata alluded to, Kaijin has a large curation team where we are continually going through the literature and capturing the relevant variant information found in studies. And in addition to that, we incorporate multiple third-party databases to help build our understanding. And so within the software itself, when we run, in this case, a particular sample from our cohort through Qiagen Clinical Insights Interpret Translational, what we are going to get back is a table of filtered and prioritized variants. And in particular here, one useful feature within the software is that we can take all of the evidence that we find for a variant from the literature curation, from third-party databases, from different functional effect calculations, and actually utilize ACMG-based guidelines to compute a pathogenicity classification. So within this table itself, that will be a result for a sample analysis, we can see here that the MLH1 gene, the NBN gene in particular, are red. That denotes a classification of pathogenicity, whereas we might see other variants that are white, that are unknown significance, and then green, perhaps benign. Now, for each variant, we'll also have a table of annotation. And very nicely within this table, since we're looking at germline data, we would be able to see, are any of these variants observed within the HGMD database as well. Now, from this table itself, for any variant of interest, what we'd have the ability to do is essentially come in and look at the different pieces of criteria that were used with the ACMG guidelines to essentially generate a classification of this variant being pathogenic. And so again, here we're focusing on what I showed in CLC, that deletion in the MLH1 gene. And we can see there are various criteria that have varying degrees of strength that would suggest that this variant in these samples is pathogenic. Now, along with this classification, what we will also generate is the bibliographic information as well. So all of the curated literature that we have for this particular variant of interest. And I'll actually go through this in a little more detail within the software. But the final thing that I want to just showcase to everybody is very nicely when, in this context, we're talking about germline variants, we have the ability to link out to HGMD, and through kind of the table we saw earlier, we'll be able to get at the full set of information found within HGMD as well. So what I'd like to do now is actually go ahead and show everybody how we interrogate this within the software. So I've just opened up Qiagen Clinical Insights Interpret Translational, and this is a web-based tool. So I've already gone ahead and analyzed my VCF file from a sample of interest. This is the LS9 sample from the study. And the analysis itself is very simple. It takes a couple of minutes to set up and run. Once it's run, our key set of results, again, at the bottom of the screen here, represent this table of variants that have been filtered and prioritized. So for each variant, we'll have a row of information with the C dot, P dot nomenclature, the phenotype that's most associated based on the information we've curated in the knowledge base. You can see there are a number of variants here associated with Lynch syndrome. We'll have its most common mode of inheritance, again, defined by what we know in the literature. We'll have information relating to the functional impact. We can see here that this variant, I'll focus on the GT deletion. That's a frame shift mutation. We also have another pathogenic mutation in NBN, and that's a stop gain. We'll also have other information related to kind of impact prediction. The CAD score here we can see is very high for these two pathogenic variants. We can also look at population frequencies across variant databases. And again, lastly here, we'll have the HGMD information in the final column. Now, in this context, for any variant of interest, what we would have the ability to do is one of two things. As we select a variant in the table, what will happen is you'll notice the top of the screen changes. And within the top of the screen in particular, we'll have, again, variant-specific details towards the left-hand side. And towards the right, we can see the computed classification based on ACMG guidelines. So here I can quickly look at the evidence for pathogenicity and also against pathogenicity for a given variant of interest. In this context, though, oftentimes I might want to look at this information in further detail. And to do so, once I have a variant selected, I can click Open. And now I can look at the explicit criteria that were utilized to establish that this variant, in fact, is a pathogenic variant. For each of these pieces of evidence, we have an associated criteria ID, a strength, and for those pieces of evidence that tie into the literature, we'll also quickly be able to link out to a bibliography that has the associated reference information. Now, as I mentioned, the knowledge base also focuses on curating literature as well as incorporating third-party database information. So for any variant that we identify, we can actually get at the full spectrum of literature that's been captured in relation to this variant simply by clicking this View Bibliography button. And what I want to highlight here very quickly is that we can filter, let's say, the results based on different categories of studies of interest. And very nicely here, just to show you kind of the frequency and the nature of updating our knowledge base and our literature curation efforts. This second article here actually is the article from which we've captured these Lynch syndrome samples. So that shows up. We've curated that through our efforts. And then also very nicely, we actually have even a more recent publication. So this is from June of this year that also identifies this variant in a slightly different context. And for any of these, we can very nicely link out to PubMed and interrogate the article in further detail. So overall, when we are in the context of interrogating a single sample, what Qiagen Clinical Insights Interpret Translational allows us to do is very nicely go through a process where we can take in a VCF file, we can utilize a rich set of information in the knowledge base and help us prioritize those variants that may be pathogenic in our given study of interest or our sample of interest. Now, so far we've looked at this in the context of a singular sample. But what I do want to note is that also in our study, remember, we are dealing with a cohort of 84 samples with Lynch syndrome. So the software also provides a mechanism not only to identify samples one at a time, but also analyze a cohort of samples. And so what we're looking at in the slide here is actually that analysis looking across all 84 samples. And we've set it up to identify those variants that appear in 2 or more individuals within this cohort. So really this table itself that we're looking at in this slide is very similar to what we've seen already in the software. The key difference here is that under this cases column, we do flag up how many samples with the black line have a particular variant of interest. And here we can see across the cohort of 84 samples, not only do we see variants in MLH1 in particular that are recurrent, but also MSH2, again, 2 genes notably associated with mismatch repair. So really at this point then, just to highlight how we process this cohort of samples, we have a filtering cascade that allows us to set different criteria to hard filter out variants of interest based on different criteria. I'll go over that in a minute. And so let's go ahead and quickly jump back into the software. I'm now going to switch my results over to those from the full cohort of 84 samples. And again, the results from this once loaded will look very similar. The key difference here with the table at the bottom is that for each variant, now we can mouse over these black bars and see which individual samples show that given variant of interest. And in addition to that, again, just like with the initial analysis for any variant, I can open up the details, I can view the bibliography, I can link out, for instance, to HGMD, get the information there. So it's very flexible. But in the context of analyzing a cohort of samples, what might be relevant is to actually do some filtering steps. So we have this customizable filtering cascade that will allow us to filter variants based on confidence, based on how common they are in the general population, characteristics related to the deleterious nature of a given variant, and also just characteristics on the cohort. So again, as I mentioned, I filtered this down to identify those variants that are pathogenic or likely pathogenic that show recurrence in at least 2 out of the 84 samples. So really this filtering cascade, you can come into any section here. You can see here that I've selected this for 2 samples out of the 84 in the filter. And as we go down this list, what we can see is using what I've defined as a filtering cascade, we've gone from a total of over 1,000 variants down to 15 potentially pathogenic and likely pathogenic variants of interest. So at this stage, what we can do now is actually take this information that we've gained. We've generated a set of pathogenic, likely pathogenic variants in the cohort as a whole, and we can export this information in a number of ways. So if you direct your attention to the right-hand side of the screen, there is an export button. So for any cohort or sample-based analysis, we can export a VCF, or we can export tabular data. We can include any number of information in columns relating to that variant itself. You can see here just general positional information, different predictions from SIFT, polyphen, CAD, as we talked about earlier, and then also annotation with different variant databases like HGMD, as we talked about, Cosmic, OMIM, just to name a few. But also from the screen, what we would have the ability to do is actually export this data into Ingenuity pathway analysis. And the goal here is to essentially take the set of gain or loss of function variants within genes found within this cohort and try to do some biological interpretation of what gain or loss of function means for this specific gene set. So at this point, this takes us to the final aspect of today's talk, which is discovery. And so what we're looking at here are kind of the end results of the analysis that I did on this set of gain or loss of function variants in the cohort of Lynch syndrome patients, where essentially we're looking at a heat map here. And within this heat map, we have our set of genes. This first column here represents the cohort itself of my study. And within this cohort, we can see each gene has a green colored cell that represents a loss of function in the set of genes here. Again, MLH1, a lot of FANC family of genes. We can see BRCA2 as well. And then in addition to this first column, we see a number of additional columns. And what these additional columns represent are actually gene expression studies from over 90,000 public studies that we have curated and pre-analyzed in IPA. And the studies that are specifically within this heat map represent those studies in which we see at the gene expression level a similar subset of this set of genes in my loss of function study that show a decrease in expression that would have a similar kind of activity impact than would our loss of function variants. And we can interrogate the contextual details of all these different public studies to understand, where else is this signature of loss of function identified across a large spectrum of different public studies related to cancer. So how we actually got to this point is that we utilize what we call the core analysis within Ingenuity Pathway Analysis. So much like Qiagen Clinical Insights Interpret Translational, Ingenuity Pathway Analysis relies on the Qiagen's knowledge base, relies on a set of over 8 million findings, and where our curation team has manually curated the literature to understand molecular interactions and associations, as well as the contextual details of those interactions and associations. And so with this data set, when we have a set of variants with loss of function, we have the ability to understand the genes for which these loss of function variants occur in. What pathways might they be related to? And what we're looking at here is something we call networks. So taking this set of genes that have loss of function variants and asking, based on our known set of interactions for these genes from the knowledge base, what are genes that could occur in an interaction network across my set of genes? So how they might uniquely interact together in this context of this cohort of Lynch syndrome patients. So essentially, each line here represents how these two molecules would interact based on what we know from the literature. And so within this interaction network, we can actually take our set of genes that we've defined that have a likelihood of neighbors of interactions that could overlap with each other. And we can look at different functional aspects that are enriched for in this set of genes. So in this table to the right-hand side, we can see lots of kind of familial cancers that show up here. We can also see Lynch syndrome in particular, and a number of other things. But this gives us some functional understanding. Now, you'll also notice each of the molecule nodes on here is green. That represents that, in fact, these genes show a loss of function in the variant data. Now, with this information, we can actually take this pattern of loss of function in these genes and utilize what we call analysis match in IPA, employing 90,000 plus comparisons from public studies that focus on gene expression to ask the question, in what studies do we see gene signatures that show a similar loss of expression of the same set of genes I just showed in my network? And so to do this, we utilize something we call the activity plot. And within this plot, very simply, we have what we call the z-score on the x-axis. And that's our metric for similarity and dissimilarity. Very simply, values above 2 typically represent where the gene sets overlap between our variant data and these expression studies, that the pattern of expression of the genes recapitulates that loss of function in our gene set that we observe from the Lynch syndrome study. And so within this, we can select points that show a high degree of conservation in this loss of activity for our given set of genes. And when we select these points, we can get a list of studies here. We'll go into this a little detail in the software. And from this point, any of these studies, we can now ask the question, when we see this pattern of expression change overlap with our loss of function pattern, what genes show concurrent loss of function and a decrease in expression in these studies? And so this translates us back to this heat map that we saw in the beginning. So what I'd like to do now is actually take you through the process of interrogating this data, lastly within Ingenuity Pathway Analysis. To do so, I'm going to open up the software, And again, I've foregone kind of going through the steps of uploading and analyzing the data. That's already been done in advance just for time purposes. What I want to focus on are the results of the core analysis, and I have them open here for this particular data set. So we can see variant effects analysis within Lynch syndrome. This is encompassing our loss of function genes. And when we run this core analysis, we'll get a number of biological endpoints, but one critical one might be trying to understand the pathways that are associated with these set of genes that have loss of function variants. So in the summary page of this analysis, the first tab at the top left, what we'll see are top hits with regards to canonical pathways. We can see role of BRCA1 in DNA damage response, hereditary breast cancer, mismatched repair in eukaryotes, DNA double-strand break repair by homologous recombination. All different characteristics of DNA damage we associate with Lynch syndrome mutations, so that's a good kind of biological confirmation of our findings. And in fact, we can interrogate this information in further detail by simply clicking on the Canonical Pathways tab. So what we're seeing here is essentially a bar chart where the height of these bars represents the enrichment of my gene set that have loss of function variants. And you'll notice this first bar has a coloring. That is a prediction based on the loss of function of these genes that we'd have inhibition in this particular pathway. But you can see here again in this, I'll just rotate this to a horizontal view. Again, the pathways that I mentioned that we would expect perhaps to be associated with mismatch repair mutations in Lynch syndrome, BRCA1 in DNA damage response. You can see mismatch repair in eukaryotes, and then also DNA double-strand break repair by homologous recombination. And non-homologous recombination, the nucleotide excision repair pathway, right? All logical pathways in the context of our phenotype of interest. Now, for any one of these pathways, we can simply click on the bar, and in the associated table at the bottom, what we'll see are the set of genes from our data set that have the loss of function variants that fall within, again, the pathway that I have selected here, role of BRCA1 in DNA damage response. So this table will give us some general annotation about these genes. We can click on the gene symbols to actually pull up what we call the gene view page, all the information we have curated on a molecule, so you can learn more about that. And then also at the tail end of this table, to the far right, we have those genes that are known biomarkers and genes that are targetable by drugs, if any. So from this point, what we can do, if we want to visualize this pathway in further detail, we can actually click the Open Pathway button. We can interrogate where within the structure of this pathway we see changes or loss of function variants. And that would be represented by all the nodes in this network that show a green coloration. So these are observations in our data set. And we can actually do predictions based on these observed loss of functions very simply by going to Overlay, selecting the tool, Molecule Activity Predictor. And what we're going to do here is utilize this observed loss of function and what we know from the literature and the directionality of the interactions with these loss of function variants with other molecules in the network actually predict what we would expect the activity of other molecules in this network to be. So very simply, I'm going to click Start Prediction here. And we're utilizing information from the literature to now color the majority of these other nodes that you can see in blue here, which represents a predicted loss of activity and inhibition in these molecules. And again, this might be representative of changes at the DNA level, or sorry, not the DNA level, the protein level or the transcriptional level, for instance. Again, predictions we're doing based on the literature. But very nicely, based on these predictions, we'll also notice that specific nodes that relate to functions within this pathway are changed, including homologous recombination and ultimately DNA repair. So overall, we have a nice, efficient way in which we can interrogate the pathway context in which our loss of function variants are influencing different key canonical pathways. Now, the other aspect I want to briefly show of the software is very simply what I alluded to in my slides. It's found under Networks. And here we're utilizing an algorithm to identify for the set of molecules that we have in our signature of loss of function within Lynch syndrome, how might these molecules be composed of interaction neighborhoods? How might they interact with other molecules in our list or other key molecules perhaps not captured directly within our list? And so we use all the interaction information available in the knowledge base to actually build these networks in a data set specific fashion. And again, just like with the pathways, what we have the ability to do is visualize the associated interactions within the networks. Again, here I pull up a network. This is not a canonical pathway, but a network of interactions based on my loss of function genes. All these edges, again, represent literature evidence of interactions in some fashion between the molecules. And so I can get some context, understand with an existing understanding of the biology of Lynch syndrome, try to understand why these molecules might be interacting in a certain context. But what I can also do is look at perhaps the functional consequence or the phenotypic consequence of some of these molecules, simply by going to overlay and selecting diseases and functions. So what we're doing in this context is taking this interaction network, its set of molecules, and looking to see, is there enrichment of any functional or phenotypic category that might be relevant to my cohort of interest. So again, here we're looking at Lynch syndrome. Again, we're pulling on the knowledge base, so this will calculate. And essentially, once it completes, we can see all the categories of functions that are enriched for. And what I just want to highlight to everybody is, in particular here, we see hereditary disorders, lots of hereditary cancers show up in this list, as well as different functions related to DNA replication. And to associate the genes in my network with any of these, I can simply click, add that function to the box, and now I know what genes, let's say, are explicitly associated with repair of DNA in this network. So that's one kind of quick interrogation of this data, looking at what's in my study. But to get to our final point for today, we might now ask the question, with this observed network and the pattern of loss of activity or loss of functions of these genes in this network, do we see this signature show up in expression data in public studies? And to answer that question, we'll simply click on this button here for pattern search. This will generate the activity plot that I showed in my slides. Again, here, the key takeaway is that the points that fall to the far right represent gene expression data sets that show the same set of genes with decreased levels of expression, and points to the left, which is opposite of our pattern of loss of function. These are data sets in which we'd expect increased expression of our set of genes. Very simply here, I can grab, let's say, all the studies that show a value above 2, again, a confident prediction in a match. And as I select these points, what I will generate at the bottom here is a table of all the studies, again, that show conservation of the signature and decreased expression of these loss of function variant containing genes. So within this table, lots of different characteristics that capture, essentially to the right here, the case and control criteria that are used in the comparison, as well as details about the overall comparison. And for any of these studies, as I click on the rows, to the top right here, I'll get details about the context of experiment. Here, this is a comparison between 2 treatments. It's looking at pancreatic cancer. It's done on NGS. Here are attributes with a control sample, so epithelial cells, the case samples, and also a link to the PubMed study itself, and also a link to the GEO page for this publication. So this data set has essentially been pre-analyzed in IPA, and we have this for over 90,000 public study comparisons. Now, I can then kind of go through this list. There are many kind of scenarios where we see similarity, but one overriding theme that I observed when looking at this is that many of these studies show treatments with some kind of compound that induces DNA damage. So kind of makes sense, and again, most of these are cancer-related studies. Now, the last step here would be to select any number of studies that I deem relevant I can just select them within the table. And now open up this gene heat map to ask the question. We see conservation of this loss of function pattern in our data set with regards to gene expression in public studies. Now, what specific genes whose loss of function are identified in my data set in network 1 in the first column of this table actually show decreased expression in any of these studies? And what we'll generate is this heat map based view where as I scroll down, I'll start to see a number of genes within a cluster here that would say that they show MLH1, again, a lot of the FANC family of genes, BRCA2, those all have loss of function variants in my Lynch syndrome cohort. And very interestingly, across these studies, again, different cancers treated with DNA damaging agents, we see that these genes, in fact, show a decrease in expression. So the green cells across each of these different columns. So now I have some insights and I can explore this further. When we talk about public comparisons, we have lots of different comparisons available in the context of, as we're looking at here, many treatment-based studies, but also disease versus normal, responders versus non-responders. But hopefully this represents a way in which we can integrate loss of function information from a variant-based study to gene expression patterns within public data sets. So with that, I'd just like to conclude. I'd like to mention again, everything that we've looked at today really falls under our Qiagen Discovery Insights portfolio. We talked about earlier using the Qiagen CLC Genomics Workbench to move from FASTQ to identification of variants. And then for interpretation purposes, we talked about at the variant level, Qiagen Clinical Insights Interpret Translational and Ingenuity Pathway Analysis. And ultimately, hopefully what I've shown everyone today is that we've walked through the step of going from FASTQ files using CLC Genomics Workbench to analyze and call variants that are accurate and with confidence. From there, using Qiagen Clinical Insights Interpret Translational to prioritize those variants both within samples and across a cohort that are pathogenic and in this case represent a loss of function of a given gene set of interest. And then lastly, utilize Ingenuity Pathway Analysis for discovery, taking that gene set with a loss of function variance and determining what pathways are relevant. How does this loss of function signature relate to gene expression signatures in various public cancer studies? So with that, I'd like to thank everybody for listening in today. And at this point, I will open it up to any questions that you may have.
Great. Thank you so much, Eric. That was a great presentation. Thanks for walking us through all those steps and putting those pieces together. Again, last chance to ask some questions here today. We've got a few questions here in the queue. Let's start off with this first one. Can we use it to check somatic variations for tumor samples without the normal reference? I have NGS sequence data available for only 40 tumor samples with no normal data.
Yes. So in the Genomics Workbench, we have pre-built pipelines for workflows that can run in either of two contexts for somatic data. So to your scenario, if you, let's say, are missing a matched normal, you can call variants, and perhaps you could employ a filtering step where you look at those variant alleles that are in dbSNP. You could filter them out to perhaps remove germline variants, or you can look at variants that have expected kind of allele frequencies, 50% for heterozygotes, 100% for homozygotes. You can filter those out as well if you choose and try to get at those variants within a sample without a match control that are somatic variants. And then conversely, we do have kind of workflows in place if you would in the future have paired tumor normal data, how you can filter out the observed variants in the normal from the tumor samples.
Got it. Great. Thank you. Another question here. Does Qiagen Clinical Insights perform analysis on microbes for virulence or metabolic genes, for example?
So currently, the curation efforts for the knowledge base are focusing on human data sets. So essentially, it would fall under the context of two main areas: hereditary disorders, looking at human germline data that we curate from the literature and through third-party databases, and also then somatic changes. So again, all of this is in the context of human data specifically for variants. So the short answer would be no. In the context of at least CLC Genomics Workbench, it definitely has the flexibility to at least call and look at, let's say, amino acid changes in microbial variants in that context. So for the interpretation aspect, again, primarily focused on human hereditary and somatic diseases.
Got it. Does Ingenuity generate PCA plots?
It does not. So really, Ingenuity pathway analysis is built for interpretation. So if you wanted to, let's say, do a principal component analysis, there are abilities to do that, specifically in the context of gene expression data within CLC Genomics Workbench.
Yeah. Okay, great. And then looks like maybe the last question. For heat maps, does Ingenuity have the ability to cluster according to different metrics, Euclidean, Manhattan, etc.?
So currently the clustering, I didn't show that with the heat map that I showed. We do have clustering based on different criteria. I think there is one option, I can't recall off the top of my head, for hierarchical clustering. So it's just one of those metrics, I believe Euclidean by default. And then there are also other metrics based on patterns that you might expect across studies, or basically scoring of different categories. But for hierarchical clustering, there's only one metric that's used when we employ that in IPA explicitly.
Yeah, got it. Actually, last question looks like that just came in. Can we analyze NGS sequence data directly in IPA, or is it complementary to the Qiagen CLC Workbench?
So it is complementary to the Workbench. With Ingenuity pathway analysis, as you saw today, we could bring in a list of genes with gain or loss of function variants. And more commonly, as you saw through the public data sets, the typical starting point in IPA would not be raw sequencing data or even quantified expression in samples, but rather differential expression in your particular study.
Yeah. I think the best way to think of that is that your CLC Genomics Workbench is for your secondary analysis, which gets you to that differential expression, and then IPA takes it over to take that differential expression, and we'll just use that term broadly. It could be, protein expression, obviously mRNA expression, microRNA, et cetera, and then analyze it there in IPA. So with that, we are at time. Thank you so much. Eric, are you still with me? Oh, never mind. We did answer this question. I thought we had a last minute question, but no, we took care of it. So with that, I'm going to say thank you and goodbye to everybody. That concludes today's session. Bye.